Alright, so I'm going to be doing my presentation on asthma. Um, so, as far as my interpretation of asthma prior to working in my master's, I would say it was pretty basic understanding. I knew it was a condition where the airways narrow and swell and produce extra mucus from either environmental and or genetic factors, which would trigger their attacks. Some examples of triggers would be pollen, infections, cold air, exercise, um, and they cause symptoms like shortness of breath, wheezing, chest pain, etc. And for some people, asthma is very minor, and others it could be life-threatening. I'm seeing some of the most extreme cases, and it definitely could be fatal. Um, and also, we know that asthma cannot be cured, but its symptoms can be controlled via medications. And then I just kind of drew out a little picture showing like a normal airway compared to an asthmatic during an attack. Um, and you can kind of see that the walls are thickened. There's more mucus, tight and smooth muscles. There's a lot of air trapping going on compared to a normal airway. So now to talk on a more molecular and graduate level. I'm going to be talking about asthma and a couple of the cell types that cause asthma symptoms, which are epithelial cells that cause airway inflammation, excess mucus, and airway narrowing. Inhaled stimuli cause epithelial cells to secrete the cytokines, TSLP, interleukin 25, IL-33, which act on subepithelial dendritic cells, mast cells, and innate lymphoid cells to recruit hematopoietic cells and cause the release of T helper 2 or Th2 cytokines. Stimuli also activate afferent nerves in the airway epithelial that can release peptide mediators and trigger reflex release of acetylcholine from efferent fibers in the vagus nerve. This response gets bigger when there's also subsets of T cells that secrete cytokines and that act on epithelial and smooth muscle cells and drive symptoms. So in this picture, we're talking about the cell communication in the airway wall of an asthmatic patient. The environmental triggers act on airway afferent nerves and airway epithelial cells to initiate responses in the multiple cell types that really contribute to mucus metaplasia and airway smooth muscle contraction. So the epithelial cells release TSLP and IL-33, which act on airway dendritic cells, and IL-25, which together with IL-33 act on the mast cells, basal cells, and innate type 2 lymphocytes. These secreted products stimulate dendritic cell maturation that facilitates the generation of effector T cells and triggers the release of both the direct bronchoconstrictors and Th2 cytokines from the innate immune cells. And that feeds back on both the epithelial and airway muscle, which further facilitates amplification of airway inflammation through the subsequent adaptive T cell responses. So as far as the cell biology of airway epithelium, they're ciliated and circulatory cell types, and they maintain the mucociliary action, which is important for airway patency and defense against the pathogens. Cilia covered with mucins and mucopolysaccharides that exclude mucus from the periciliar space and enhance formation of mucus layer. Circulatory cells create a class of mucins, the polymeric gel-forming mucins. The two major airway gel-forming mucins are MUC5AC and MUC5B. Some secretory cells like mucus or goblet cells produce mucins and store them within mucin granules, but other cells produce this and secrete mucins but lack prominent granules. Gel-forming mucins are secreted to the airway lumen and create viscoelastic properties of the mucus gel layer. As far as airway epithelial injury and remodeling in asthmatic, these changes include airway wall thickening, epithelial hypertrophy, mucus metaplasia, subepithelial fibrosis, myofibrosis, hyperplasia, and smooth muscle cell hyperplasia and hypertrophy. Airway remodeling is pretty much an ongoing tissue injury caused by stimuli. Signs of injury are loss of epithelial integrity, disruption of tight junctions, impairment of barrier function and cell death, mucous metaplasia, and subepithelial fibrosis. The epithelium is both a site of production of these mediators and a source of cells that respond to mediators produced by the immune cells and other cells within the airway. As far as the airway epithelial, how it contributes to the Th2 responses, the Th2 cytokines play a huge role in asthma and the TSLP. GM, CSF, IL-1, IL-25, and IL-33 are made by the epithelium and create the Th2 cytokines. There's an interleukin-13, 33, and TSLP in asthma, and IL-13 is made by the innate lymphoid cells. IL-33 causes changes in airway epithelium. mRNA and miRNA expression patterns in airway epithelial cells. The IL-13 induced protein periostin is secreted basally from airway epithelial cells and can be used as a biomarker for Th2 asthma. And half asthmatics are Th2 high and respond really well to corticosteroids and anti-interleukin-13 antibodies. As far as mucous metaplasia, severe asthmatics, as we know, which obstructs airflow, and this is obviously because of increased mucin production, secretion, and changes in mucin cross-linking, mucus gel hydration, and mucus clearance. Mucous metaplasia results from more production and storage of mucins and secretory cells, and lots of different stimuli and signaling pathways regulate mucin production and secretion in the airway epithelial cells. So the interleukin-13 on airway epithelial cells causes the mucous metaplasia, and patients with Th2 high asthma have high levels of bronchial epithelial cell MUC5AC mRNN compared with healthy patients with Th2 low asthma. And a study showed that MUC5AC helps with clearance of enteric nematode infections and protects against influenza infections. An increased MUC5AC is a part of the immune response that contributes to host defense against pathogens. And the fact that MUC5B is required for normal mucociliary clearance and defense against airway infection tells me that we should pay attention to decreases in our MUC5B because it is a big contributor to the airway dysfunction in asthmatics. And IL-13 is recognized by surface receptors, including airway epithelial cells. And the airway epithelial cell IL-13 receptor that is important for mucous metaplasia is a heterodimer made up of IL-13 RA1 and IL-4 RA. So as far as this picture, kind of talking about how IL-13, and we're talking about IL-13 induced mucous metaplasia, by the way. So IL-13 binds to its receptors on the surface of the mucous cell progenitors, leading to phosphorylation of the STAT6 and translocation of STAT6 heterodimers to the nucleus, where they bind to promoters of STAT6 responsive genes. STAT6 dependent processes that contribute to mucous metaplasia include a CLCA5 or a CLCA1 dependent pathway, a SERPIN dependent pathway, and a 15-lipoxygenase 1 dependent pathway. And the transcription factor SBDEF is a master regulator of mucous cell differentiation, inhibits the FOXA2, which represses mucous cell differentiation, and activates transcription of other genes that are expressed in mucous cells. In the secretory pathway in the mucous cells, mucin monomers are pretty big proteins that need a lot of processing in the endoplasmic reticulum and Golgi, and each mucin monomer contains about 200 cysteine residues that can help the intra and intermolecular disulfide bonds. And the ER of the endoplasmic reticulum of mucous cells has special molecules that are required for the processing of mucins, and one of these is the anterior gradient 2 molecule, and an active site cysteine residue in AGR2, which forms mixed disulfide bonds with mucins. In a study, they found allergic asthma AGR2 deficient mites had less mucus production, and that's associated with activation of the unfolded protein response. And it can have a direct role in mucin folding or another function necessary for maintaining normal function of the mucus cell ER. And as far as the ciliate cell structure and functions in asthma, to really quickly hit on the ciliate cells, if there are abnormalities, it really does cause or mean severe asthma, because it means increased numbers of dead cells and loss of epithelial structure, which means that the ciliary dysfunction may be a consequence of the epithelial injury, which causes impaired mucociliary clearance. And I kind of want to talk about a little bit of the cell biology of airway, smooth muscle, and asthma. So most of us relate asthma to narrow airways, as I talked about in the beginning, which leads to shortness of breath, respiratory failure, potentially be fatal. And it's really due to the contraction of the bands of the smooth muscles present in the walls of the conducting airways in the lung. Contraction of smooth muscle can be physiologically induced by release of acetylcholine from efferent parasympathetic nerves, or by release of histamine and cystine leukotrienes from mast cells and basophils that causes that airway narrowing. And the physiological responses to release acetylcholine from efferent nerves or release of histamine and leukotrienes from mast cells and basophils cause only mild airway narrowing in healthy lungs. But in asthmatics, they have a marked increase in sensitivity to the agonist that can cause that airway resistance and drops in maximal expiratory flow rates. And the studies make it pretty obvious that all allergic humans release similar amounts of bronchoconstrictors into the airways. But only asthmatics develop the exaggerated airway narrowing. Talk about a little bit of the mechanisms regulating generation of force by airway smooth muscle, actin, myosin coupling. So the force generation by airway smooth muscle is mediated by interactions between actin and myosin that depend on phosphorylation of the myosin light chain by the serine threonine kinase, myosin light chain kinase. This process is really negatively regulated by myosin phosphatase. Increases in intracellular calcium concentration as muscle causes contraction by two parallel pathways, one bound to calcium, the serine threonine kinase calmodulin directly phosphorylates, and that activates myosin light chain kinase. Increased calcium also increases the GTP loading of the GTPase RHOA, which increase the activity of its downstream effector kinases and RHO-associated coil-containing protein kinases 1 and 2, and which is also labeled ROC1 and 2. And ROCs directly phosphorylate myosin light chain phosphatase and affect that inactivates the phosphatase, further enhancing myosin phosphorylation. RHOA and it can also be activated independently of increases in intracellular calcium. So just really to kind of talk about this picture that I drew, kind of showing the core signaling pathways that are responsible for airway smooth muscle contraction. So airway smooth muscle contractile forces is really generated by cyclic cross-bridging of actin and smooth muscle myosin, which depends on myosin phosphorylation. Myosin phosphorylation is regulated by the cyclic increases in systolic calcium that activate the calmodulin to phosphorylate myosin light chain kinase, which directly phosphorylates myosin. In parallel, the small GTPase RHOA is activated by both calcium-dependent and independent pathways. RHO directly activates RHO-associated coil protein kinase, which is labeled as ROC, and which in turn will phosphorylates and inactivates the myosin light chain phosphatase, which is labeled MLCP, which normally will actually dephosphorylate the myosin. And most importantly, the physiological pathway for increasing the cytosolic calcium in airway smooth muscle involves activation of the GAQ by G protein coupled receptors that respond to extracellular contractile agonists, such as the methylcholine, serotonin, and histamine. And the GAQ activates phospholipase C and B, which generates IP3 to bind to IP3 receptors on the sarcoplasmic reticulum and release the sequestered calcium. And that's pretty much it. And then the next slide is just my references. Thanks.